Oh, I think we're live.
Is it working?
I think so.
Hey, all right. Can we get someone from the chat to chime in and say, yes, you can hear us? Yes, you can see us?
We did it.
Made it work.
All right. Hi, everybody. Hopefully most of you stuck with us. We were having some technical challenges with Crowdcast. I know that they've had some challenges sort of stepping up their bandwidth in response to everyone under the sun moving their content to digital, including some bigger conferences. So I'm guessing that's what may have led to some of the connectivity challenges that we had leading up to this. We were like literally in the green room and trying to push this thing live, and it repeatedly would not work. But I'm glad that we're live now, and fingers crossed that we can get through this whole thing.
Hope it keeps on working.
Exactly. I'm Richard Priest, co-founder of Escarpment Labs. Got Nate here as well, Nate Ferguson, another co-founder of Escarpment Labs. The purpose of this session today is Yeast Basics Q&A. There's been tons and tons of dialogue, tons and tons of questions in the first three sessions that we had on Yeast Basics, where we went into, what is yeast, how does yeast make flavor, and then harnessing yeast flavors. And we tried to collect a good representation of all of the questions that we were getting to answer in this presentation, and spend a little bit of time thinking about some of these questions so that we can come up with answers. And really, really happy that you guys pitched in and asked questions. It helps guide us to what you guys are interested in and what you're looking for answers for, and forces us to think a little bit about some of the questions that we didn't have very good answers for as well. So it's a learning experience for us as well, and I think that's really exciting. I also see 22 more questions at the bottom, so we will do our best at the end of the presentation to answer those questions as well, and do as much as we can. And if we have to, what we'll do is we'll push some of these to future talks. We have a number of other presentations coming up that I just wanted to mention off the top here, just because I've seen that, not a whole lot of you have signed up for these yet. So if you can, anytime during this talk or afterwards, you can just go over to our Crowdcast profile. You can click that at the top and see some of the other webinars that we have lined up. We have a webinar on beer yeast flavor diversity. So if you've ever wondered, what yeast is most similar to the Ardennes strain, or what yeast makes the most glycerol, or what yeast makes the sweetest, least hoppy beer, or whatever, or the opposite, the most bitter beer, we have some answers to those questions. We're going to be talking also on April 9th about biotransformation. So if you've ever wondered, what the hell is biotransformation, what's happening, what are the yeast doing to the hop compounds, we're going to try to answer those questions as well. And then April 14th, that's a Tuesday, we're going to have a special guest, Isneto. She used to work with us at Escarpment Labs doing research and development, and she did a lot of work with us to develop new lactobacillus strains with interesting flavors, and we're going to be talking about that road to what we call Lacto 2.0 as well, which I think is pretty cool stuff as well. And I know that a lot of you guys are already experimenting with some of these new lacto strains.
Mm-hmm. We also have a series of talks lined up after, we're currently lining up after that. So stay tuned. There will be more webinars coming up after that as well. We're not done after the 4.
Yep, I know a lot of you guys are fans of Lars Marius Garshol, the sort of the Kveik guy, the Kveik god, and I can confirm that we will have him on for a fireside chat about farmhouse brewing during that webinar series.
Yeah, it's going to be good.
Yep. So without further ado, I think we should get into our Q&A slides.
Yeah, here we'll pull this up.
And I do want everyone, just because we've been doing this and it's been a lot of fun, just let us know what you're drinking. Today I've got a True Believer from our friends at Short Finger Brewing in Kitchener. They started doing some beer deliveries here in Guelph, so I pounced on that opportunity and made sure to pick up some growlers from these guys. Rob's a great guy, runs a homebrew shop as well, and helps to make sure that brewers out there are able to get all the wonderful yeasts and ingredients that they need.
Sorry, we're just having a little bit of a... There we go. And through the power of technology. Whoa. We are live. Can everyone see that?
I think we're good.
All right. So today, just so everyone's kind of we kind of broken down today down into 5 main areas. So we have yeast stress and behavior, which Richard's going to cover the handful of questions that came in from there. We then have yeast flavor, which I'm going to cover, followed by yeast repitching, also me, and yeast propagation, finally me. And then we're going to finish the whole thing off with Richard talking about yeast nutrition. So please keep more questions coming in the comments. We're probably going to bring up more and more topics throughout the day or throughout the talk. This isn't the end of these things; it's just kind of the questions that came in so far. And with that, we're always working on new content. Also, if there is something you guys really want to see us talk about, or I know I saw some things that in the chats on Monday about wanting a yeast management talk and things like that. If you guys want us to cover those things, please let us know. Put it in the questions. Put it in the comments. Let us know any way you can, so we can make sure we see that. And with that, Richard, you can take it away.
All right, I'm going to be talking about yeast stress. The first question that we got was actually about something we don't sell. That's dry yeast. And the question was dry yeast rehydrate or not? I thought this was still an important question to answer because it is a common question that people have when they're brewing. And especially it's important always to try to flag some of these preconceived notions or common wisdom that may not be right. Right? For years and years and years, yeast companies that produce and sell the dry yeasts have told people, you have to rehydrate the yeast. Here's the instructions. You have to put your yeast into the warm water, give it a stir, let it sit, do all these things without really necessarily having a lot of evidence for why that was important. So some very clever people at Fermentis decided to sort of put that to the test, right? And do some research and compared rehydrated yeast to yeast that was just basically sprinkled in the wort. And what they found, and they did a pretty thorough study on this, they found across the board it really didn't have an impact. That the dry yeast, regardless of whether it was rehydrated in water or whether it was just sprinkled into the wort, didn't really change how it behaved in terms of fermentation or how it behaved in terms of flavor profile. So just to answer the question, with dry yeast, especially the ones that are made for beer, there doesn't seem to be much support for the need to rehydrate them. You can likely just sprinkle those right into the wort. And part of that is that the dry yeast manufacturers do go to their way to make sure that those dry yeasts are supplied with an adequate sterol reserve. We talked about how oxygen helps the yeast to build and incorporate sterols in their cell membranes so that they can make more yeasts in the future, and that oxygen is required for this. What the dry yeast manufacturers do is they often have processes that build sterols, extra sterols into the yeast, and so they avoid some of those issues with oxygenation, also some of those issues that might otherwise face the yeast as it goes from this little shriveled up raisin to a plump hydrated yeast cell. So these are big companies that are very clever, and they've figured out a lot of ways to eliminate this issue.
So if I add one thing to this, I know that we know the guys from Fermentis quite well. I know other companies have not tested their product as well. So if you do, I'm not going to say names or things like that, but I would still follow what they are. I know Fermentis has that certified, easy to use standard because they've gone through all of this. So I would still probably recommend. I'd still recommend what you do, whatever the pack says.
Mm-hmm. Yeah. Next slide.
All right. Yep.
You're gonna get sciencey answers from me. This is one of them.
I try to bring things a bit more approachable.
Oh, I think. Well, yeah. We can make this approachable for sure. The question was, can I use wine yeast for beer and vice versa? Is there a big difference between those yeasts? This is something that I think is an interesting question, right? Because we think of wine yeast and beer yeast as very different things, and in a lot of ways they are. And there's modern scientific evidence that demonstrates that. So if you look on the right-hand side, that's like a simplified version of the yeast family tree. We showed that, I think, a couple times in the first 3 sessions to show, what the yeast family tree looks like and how these things are related. One thing we see is that there is actually a group of beer yeast that's really closely related to wine yeasts, and that's interesting because they may actually share a lot of similar properties, like, for example, some of the same stress resistance. But most of the beer yeasts are not as closely related to the wine yeast, and that's resulted in there being genetic differences between these yeasts. So, for example, wine yeasts have adaptation for sulfite tolerance, right? Winemakers add sulfites to sort of keep all the other microbial beasts at bay, whereas beer typically doesn't have a high sulfite content. Wine yeasts often, a preferred trait is that they're killer positive, meaning that they can also secrete these proteins that actually can keep other yeasts at bay and prevent them from growing or kill them. And otherwise, they're often specialized for being able to survive in nature a little bit better than the beer yeasts. When we go and look at the beer yeast, we see that they've adapted in different ways. They've adapted to beer, right? They can consume the maltotriose, which is one of those major malt sugars that we talked about earlier, and they can flocculate. This is really important for a brewer because a brewer needs to turn a beer around really quickly, right? They're making as many beers as they possibly can in the span of a year, whereas a winemaker, they're fermenting once. That's it, right? Winemakers don't really care that much about flocculation because they have a whole year. So that makes a very big difference and kind of informs how the properties of these yeasts and explains why they're different. So to answer the question, can you use wine yeast in beer? You can get really interesting flavors because some of these wine yeasts are really, really aromatic. They can do a lot of cool things. That being said, a lot of them don't ferment the malt sugars. So you may need to finish that with a beer yeast or Brett or something else that can consume the complex sugars, because wine yeast will often finish a standard gravity beer at around 1020. Like it'll be pretty high final gravity and pretty sweet if you're only using the wine yeast. So it's really helpful to co-pitch or sequentially pitch with something else. The other question go ahead.
So the only thing I would add, and you kind of brought it up, is that the only time I see brewers using wine yeast in brewing is when they're trying to do Brett stuff, when they're trying to use something that isn't intentionally going to under attenuate the beer so that it's, so that it gives more food available for Brett. Yep.
There's a lot of potential there. And then the other question of beer yeast in wine, I think that may be a less common thing. The only thing I would add there is make sure you're supplying nutrients. Beer yeast are pretty high nutrient requiring yeasts. They're very used to being pampered, as opposed to wine yeasts, which are almost closer to wild yeast in the sense that they can they're almost a little hardier. They can survive in the wild a little bit better. The other part is watch the alcohol tolerance of the yeast. Once again, wine yeast are typically a little bit more alcohol tolerant, right? These things need to get up to, in some cases, 14, 15, 16% alcohol, whereas beer yeasts typically don't. And then, of course, watch the sulfites as well, where beer yeast might be more sensitive to those sulfites. So I will say, even just speaking from experience, I do every so often get a chance to make wine, and that's a lot of fun. And there's a few that I have fermented with Saison yeast, and specifically our Saison Maison strain, because it's really aromatic. So that's a nice yeast that can be used, for example, in white wines, where it does seem to help enhance some of those aromatic properties. So that's certainly possible as well. You just sort of have to watch the limits of the beer yeast and recognize that in some ways they are a little bit less hardy than the wine yeasts.
Yeah, sounds good.
Cool. Next slide. Love this question. I previously learned that yeast does not perform aerobic respiration, and can you discuss this? So I wish I had gone back and sort of asked this person to clarify, but I think that this question has to do with the fact that S. cerevisiae yeast, they always ferment and never go fully aerobic. Like if you give Saccharomyces cerevisiae yeast all the oxygen in the world, all the sugar in the world, for some weird reason, it still ferments. Even though
it's probably survival.
Yeah, exactly. It still ferments. We know that we're going to get a lot of ATP out of respiration, and we're not going to get as much out of fermentation, but they still do it, which is kind of an interesting conundrum. So this is called the Crabtree effect, where with a lot of oxygen, S. cerevisiae yeast, they still ferment some of the sugar into ethanol instead of using it to make biomass. And the reason for this is that ethanol production is competitive. It helps prevent other guys from growing, right? If I'm S. cerevisiae and I can make alcohol really quickly, it's advantageous for me to make a little bit of alcohol to prevent all of the other molds and bacteria from being able to grow, so that I can sort of dominate the environment and have a better chance of surviving and making more of me. So yeast use this kind of strategy. They can produce the ethanol. They can also consume it later. Like that's something to recognize, is that if a yeast has access to, especially if it still has access to oxygen, it can get some energy out of ethanol. It can turn it into things like acetic acid, for example. and get a little bit of energy out of that. So yeasts like Saccharomyces and also like Brettanomyces use this make, accumulate, consume strategy where they can make a lot of a metabolite like ethanol to ward off everyone else. They can sort of hoard it for a little bit and then consume it as they need it to sort of keep the culture going. One thing I thought was really cool is that this is also the exact same phenomenon. It's called the Warburg effect in human cells. And this is really, really well studied and really important because this is what cancer cells do to survive. Cancer cells form tumors, and a tumor environment is really low in oxygen. So the cell needs to adapt and find other ways to survive. And so basically fermenting while also respiring is the exact same things that cancer cells do. So in case you've ever wondered what's similar between yeast cells and cancer, I guess that's your answer. I think it's kind of cool that different cells from totally different organisms have found the same strategies to adapt. You know, the next slide.
Yeah.
Looks like we lost half of your faces in the bottom of the slides.
Not sure what's going on there.
Oh man.
We'll keep going. We'll keep pushing this along.
We'll do what we can.
Yeah.
Okay. You mentioned, oh man. Well, we can try to shift everything, but maybe that's not practical. Yeah. The next question was, you mentioned yeast consuming extra sugar is stressful. Does the amount of time a yeast cell is in contact with a high sugar concentration also influence the stress? And the answer is yes. If there's more sugar in solution, what you're subjecting the yeast to is osmotic stress, right? They have to sort of deal with the concentration of solutes that are outside the cell and pump a lot of stuff back and forth in order to keep the cell functioning. And so if a yeast is exposed to high osmotic stress for a long time, it can impact viability. And it's a pretty linear response in terms of the osmotic stress to the cell viability. So this is just some data pulled out of a paper from some yeast researchers, sort of showing that impact, where if a yeast is subjective, 50% sugar equivalent, they're not going to be very happy, right? And that's why, for example, you're not going to really find yeast doing much in honey until it's diluted down.
The only thing I would add to this is that this element of osmotic pressure is the same reason why jam and preserves, or one of the main reasons why jam and preserves work, is you're making it so osmotic, so thick and viscous that the substance is unable to be consumed by other organisms. Same thing goes with yeast. Yeast is a fungi, and that's the reason why molds can't grow on jam. It's the same basic fundamental principle.
Absolutely. Oh, can you go back?
Oh, sorry.
Oh, no, sorry. Yeah, go ahead. Just lost my train of thought. I'm not going to make you memorize this, but I thought this was interesting because there has been a lot of research on this topic, especially when it comes to people making fermented things like ice wines, where the starting sugar concentration is super, super high. So the professor who essentially was the yeast research mentor for Nate and myself, George Vandermerve, has published quite a lot on this topic. So I think we've gleaned a little bit of this knowledge from him, but I just wanted to highlight the fact, this is just from one of his papers, to highlight that the yeast response to high amounts of sugar is pretty complex and involves the whole metabolism changing and all of the genes that control it adjusting to the new environment, and that's kind of cool. It gets pretty complicated.
There's a lot going on.
How does yeast biofilm affect equipment's CIP capability, and what are your recommendations for cleaning, sanitizing procedures for equipment that has come into contact with yeast that can produce a biofilm? I wanted to say, like, beer stone is more of an issue than biofilms when it comes to cleaning yeast residues. Like, even for the yeast that make films, like Brett will make like a pellicle, that's typically pretty easy to get rid of. One thing that's not easy to get rid of is beer stone. So that's the inorganic calcium oxalate protein deposits that are produced as a byproduct of fermentation, especially when you have more mineral-rich water, right? If you have higher calcium content, this can be more of an issue. So for us here in Guelph, where our water is hard as a rock, this is something that we struggle with constantly. So for the pro brewers out there, I would recommend using a caustic and hydrogen peroxide mixture. You can essentially dose hydrogen peroxide into your caustic loop while it's running, and that basically serves as an accelerant to really help create more turbulence and help to remove some of those protein deposits and loosen them up. And then for anyone who really wants to dive deep into brewery cleaning and improve that, and I really hope that's every pro brewer out there wants to learn more about cleaning, because there's always more to learn. The Master Brewer's book by Richard Wrench is a pretty good one to start, and I think we learned quite a bit from that.
Yep.
And then go ahead.
No, I'll finish. I'll
And then for homebrewers, the one thing I'll say is, like, you guys have the luxury of being able to take almost everything apart and scrub it, and that's always going to be very effective. So, you guys are lucky.
Yeah. Just to give everyone kind of an idea, there have been breweries who essentially get rid of kegs because beer stone develops inside the keg, and then biofilms build up inside the beer stone inside the keg. So that keg of beer, whatever goes into that keg is always fouled, and it can be, oftentimes you can have non-biofilm producing organisms just kind of get into the remnants of previous beer stone and other organisms that are there and live there. So like you can have all sorts of different organisms get inside these kegs and essentially become consistent problems for breweries. This is, if you are finding yourself in that situation, the hydrogen peroxide plus caustic thing is a phenomenal solution. Put it in your keg washer, and then away you go, and it'll make your life a lot easier.
Yep. Can we freeze your pouches of Kveik for future use past the expiry date? Isn't Kveik invincible?
Yes. No, don't.
Well, it's close. The answer is we haven't necessarily tried with the liquid stuff. We would suggest drying the yeast first, just because if you take something that's liquid and you put it in a freezer, you're going to have that water turn into ice crystals. And at the small scale, the size of a yeast cell, water crystals solidifying could actually act like knives and actually dig into that cell and burst it. So it does help to remove the water first, like dry the yeast. Lots of people that are drying their Kveik at home. Here's a picture from our buddy Brian at Sweet Generous Brewing, showing Kveik being dried in a food dehydrator. And I would say that's something that we have tested. We've received farm Kveik from Norway and stored it frozen. And there's one that I've had for 2.5 years. And anytime I want to show someone, how special Kveik is, I go and I take a little chip and put it in a little bit of wort, and it's fermenting in 40 minutes. And this thing has not really slowed down very much. So I would say, at least 2 years you can store dry Kveik in the freezer, and probably longer. We've heard of, 10 years and beyond.
It's absolutely silly. The first time Richard brought Kveik inside the facility, I think we were making a barley wine or something like that. But you told me, like, I probably had another couple of hours of work to do inside the lab, and you inoculated it and said, like, hey, let me know if this thing starts fermenting. And I'm thinking, this isn't going to start fermenting until tomorrow. What are you talking about? And about an hour later, I messaged him going, like, what did you bring inside this lab? You're going to kill us all.
Yeah, it was 12% in 2 days.
Yeah.
I still have 4 bottles of that. We should get into that at some point.
After COVID.
Yeah. All right. I think that's it.
Yeah. So we'll now go in a bit more to yeast flavor. And a lot of these questions are kind of us delving a little bit deeper into some of the things that you guys want to learn more about. So the first question, how would you go about breeding flavors into your yeast? So we have to talk a little bit about how this occurs. And this really occurs into 2 different regions. So we're going to talk about artificial selection / genetic drift is the first one. And genetic drift and mutation is going to be something we talk about a few times. So keep that in mind. And the second one is yeast breeding. So to give you an idea of what we can do with yeast, I want you guys to wild mustard is a great example of this. And this is what we call artificial selection. So cauliflower, broccoli, cabbage, kale, and kohlrabi all originally came from wild mustard. They all originally came from something that doesn't look anything like the end product. So all humans have done, remember, we did this. This is all humans. The artificial means we put the pressure on. Natural means nature puts the pressure on. We put the pressure on this. We just bred this wild mustard to express a gene more frequently. It's kind of that idea, like, if you sheep and the one sheep becomes extra fluffy, well, that one's going to be the thing that breeds with and makes all the next round of sheep. or you can go for cows, or you go for yeast. You're just selecting the ones that operate the way you want them to work. Now, this isn't just for yeast and mustards. To keep the theme of dogs in this presentation, we also see this happen for almost all the dog breeds. So almost all dog breeds around the world, I think actually all dog breeds around the world, can be traced back to wolves. And we can see this in Europe, North America, China. We all had these different wolves that were then artificially selected by humans for certain properties. And that's how we got the first round of dog breeds. Those were then further pushed even further to become even more hyper-specialized into all the different dog breeds we see today. This is a great example of artificial selection. So humans applying pressures for things that we enjoy onto these yeast cells. So the same thing has happened with yeast, where if we had a fermentation that we liked the way in which that one tasted, we're going to harvest yeast from that one and then proliferate it everywhere else. We see this happen in a lot of mixed ferment facilities, where if they have a wonderful barrel, they'll take that one and try to push it everywhere they can. If you're in a brewery and you have a mixed ferment culture and you like that one barrel, you push that one everywhere. You try to spread the good as far as you possibly can. Now, we actually have a few kind of somewhat detailed history. There's a little bit of folklore in these things, a little bit of kind of mystery in it, but the basic bits are there. So Kaliel, the equivalent to your WLP001, US05. You can probably think of other versions of Kaliel, but you know what I'm talking about. It's believed to originate from a Northern English brewery. And there's a lot of things to suggest that Northern English brewery might have gotten their yeast from Kolsch via the Hanseatic Leeds trade and all sorts of other stuff. The history really falls apart once you go past there. So the things on this are a little gray, but we know that it went a little bit like this. So there's a brew pub in BC that worked at this brewery, brought it over to the brew pub in BC, and was working with it there. One of the brewers at New Albion Brewing in California were the first really American brew pub and the first brewery that made the American Pale Ale. Went to this brew pub, loved the yeast, got some, brought it back to California, started using it. The brewers at Anchor Brewing loved this beer, wanted to use it themselves, and used that for Anchor Liberty Ale. Liberty Ale's yeast strain was then either brought by a strain bank or acquired directly by Sierra Nevada. Sierra Nevada then starts using it for all their core beers, and then because Sierra Nevada has it, everyone starts using it, and it proliferated across the entire U.S. So every single one of these stages put their own little twist on it. They moved it one way or the other. They made it a bit more clean, a bit more less English, a little bit less flocculent, not quite as a brick. They turned it into their own thing. This is essentially how artificial selection works. We're just slowly moving it one step at a time into something into something that wasn't before. Slowly accumulating small little positive mutations in a sense, at least that humans select for. Now, we also have the same idea with Vermont or Conan, the kind of standard New England IPA strain. So there's a yeast lab that we know had the original version of this, which then a Vermont brew pub started using en masse. That brew pub then pushed a certain way, used it for a long period of time. There's a few top breweries that then obtained this yeast, one in particular, that used it, started turning it into their own thing, made a very popular beer, and now we have Vermont ale yeast. So a lot of these yeast strains we're working with are just pushed a little way, one generation at a time. You like the way this batch tasted, I'm going to push it further that direction. I like the way this next batch tasted, we're gonna use that one and keep on going with that kind of flavor profile. It's all kind of just small little tweaks in sequential fashion. So that's the artificial selection arm of breeding, flavor creation, things like that. The other version of this would be yeast breeding or mating. And the best example of this is lager yeast. So lager yeast is thought to be of a hybrid of Eubayanus and Saccharomyces cerevisiae. Both these, there's some mating pressure for these two to merge, which we're not going to get into today. We'll talk about that at length when we have the lager talk. But these two hybridizing had a huge amount of impact, and it changed the flavor of them massively. And the hybrid was more fit to handle the conditions than the individuals alone. And that's essentially how we had that. Now, this is a massive flavor change. This changes everything in regards to the beer. Yes, Richard loves lager yeast. And there's also, this is what we'll talk about a few webinars from now, but there's a huge amount of potential here when it comes to yeast breeding. We, along with a handful of other labs, have projects on yeast breeding that we're trying to push to get new strains out there. I will say this, it's not as easy as you may think. There's a lot of issues with it in regards to reversion and things like that, and the cells splitting back into their initial parts, which is a talk for a completely later day. But this is one way we are exploring for this to occur. One way I didn't mention here would be genetic engineering, but a lot of people frown upon that, and for arguably good reasons. So we're not going to talk about it. Richard, you have anything to add?
Nope, all good. I like lager yeast.
You do. Question 2. So trying to brew a clean lager-like beer with Voss Kveik. I have heating but no cooling capacity. Would you recommend for temperature schedule 23 pitch, then slowly ramp up to 33 at 50% attenuation? For super clean, I think you're spot on. If you were trying to go for more expressive on this, I'd probably go for the higher end, go for a 33, 34. That still feels really weird saying, but it's for the Kveik, that's exactly where you want it. The thing I would make sure to really focus on here, that is, if you want this beer to taste more like a lager, you have to treat it more like a lager. So doing things like spunding and things we talked about already kind of in passing in this series, I would recommend you do those things. One thing we haven't mentioned already that's really important for a difference between brewing a lager with a lager yeast and a lager-like beer with Kveik is that normal lagers do not possess the ability to acidify their wort as strongly as most ale yeast. So what happens, and the reason for this, why it's important, is that one of the things that makes lagers nice, light, and crisp is that the fact that they are slightly more acidic. The pH is usually at a lower than most other beers. So if I make a lager and I don't pre-acidify my wort to about 5.2, the beer ends up tasting quite flabby. If you ever have any kind of blonde ales you're making and you're finding that that's a problem, especially if you say brewing somewhere that has quite hard water, you might want to be looking at altering, like knocking your pH a little bit, adding some lactic acid or phosphoric acid. I'd recommend lactic to your mash or to your wort to try and knock that down a bit. Because Kveik has the ability to knock the pH down more than lager yeast, you don't have to do this as aggressively. And if you do it too much, it can have some other really unpleasant characteristics with it. We could go a lot more into this topic, but we are going to have a whole talk on lagers, if not just one, probably more than one. We're going to talk a lot more about this topic then.
Yep. Yeah. The only thing I would add there is that we'll get more into the pH stuff when we talk about yeast diversity. For me, having that information of which yeast drops pH which amount has really been the key to dialing in flavor with each yeast, and knowing that information ahead of time allows me to sort of dial in exactly what the wort pH should be to target a specific beer pH, and that's been really, really helpful for beers where pH really matters, like lagers and hazy IPAs. So I do think that matters quite a lot and can really help people.
Yeah, but we'll get a lot more into that at a later point. So question 3. Can you affect ester phenol production by sharp changes in temperature during fermentation? So this is kind of, to break this down, esters likely. Phenols, minimally if you can. There's some conflicting kind of concepts around that. One thing we haven't mentioned, though, this is important, is that all the esterification requires energy, it requires ATP to produce. So if you are doing sharp temperature swings, you are shocking the yeast cells and causing them to spend a lot of energy, which means they will probably divert energy away from making esters. I can't find any research on this, which we probably won't, because this is something that no brewery would be doing. I can't see any brewery wanting to do this for any confident reason, or at least, sorry, any intentional reason. I don't have much to say past that. I would not recommend you do this. If you do see an increase in phenols and esters, I would probably argue it's likely more so due to cell death and autolysis than the cells producing these compounds. Richard?
No, I agree with that. I wouldn't expect to see. I don't know if there's been much research on flavor production during temperature swings, and like part of the issue is that when we're talking science, we're usually talking about variables being controlled, right? So kind of a challenging thing. It is really hard, I think, a lot of the time to sort of model that kind of inconsistency. Because it sort of flies in the face of science, and I'm not saying that that's a bad thing or in any way. It just makes it hard to study.
It does, absolutely. Beer very much so lies in the world between practicality and research. Like you can make it very analytic, but sometimes it doesn't. I shouldn't say sometimes. More often than we'd like, it doesn't transfer into the brew house or into the real world. So This is a good question, but I would not recommend you do this, and I don't think we'll find a solid answer to this anytime soon. Now, there were a handful of questions regarding esters. So some of them were, are there specific fan constitutes that can be added to your wort to promote certain esters? Is there science and research comparing open versus closed fermentations? And then also another one, I have seen literature cite that although you potentially encourage more growth with more oxygen and more ester production, oxygen itself lowers the activity of the enzymes that are responsible for esterification. Any clarification? I was told open fermentation leads to more esters due to less dissolved CO2 and growth metabolism. We're going to talk about esters because there's a lot of things in here that are all associated with this. Now, before we go much further though on this, I need to make this really clear. Unlike almost every other flavor compound that's produced by yeast cells, Saccharomyces cerevisiae, because we switch organisms, it goes out the window. This is muddy. The esters have been frustratingly hard to figure out in regards to the science behind them. They're so multivariable, and we don't even really fully understand why the organism does this. We kind of talked about why we think it does it, which we did that in session 2, but we really don't have concrete answers here. So we can't answer some of these as fully as I would like, just because we don't know. Like the scientific community does not know, but we're going to do our best to try and answer them. So first off, one thing, I originally had this in the first presentation, but I deemed it to be too much information, so I cut it. We talked already in that all esters arise from the fusion of an acid and an alcohol. And those two merge together and esterify and become an ester. So I want us to look at the common acids and esters associated with these two, because this is where we start seeing some subclasses of esters. And what we often see become a problem is that certain things will increase one class while decreasing the other, or they'll increase one with having no impact on the other. And then if we say all esters are no esters, or in many cases, the paper is only talking about one type of ester, so the paper talks about all esters referring to that type, it gets very complicated, very confusing. So the common acids we're working with, one of the most common ones is acetic acid, or kind of its other versions of it. We then also have fatty acids, and then on top of it, just a whole myriad of kind of smaller other acids that are present that could be used. When it comes to the alcohols, we have mainly ethanol, and we also have fusel alcohols on top of that, as we talked about in lecture 2. So when we start breaking these esters down into groups, the first one is the group we call ethyl ester. And these are the most common, so these are the most common, but these are formed via ethanol. And that's where the ethyl portion of these esters come from. The ethyl means that the alcohol used was ethanol. So you use a few of these that we see in beer. Ethyl acetate is pear fruiting solvent. A lot of yeast strains produce this. One of the most iconic ones is the Duvel, or a lot of Belgian strains produce this. Ethyl caproate is apple and anise seed, and ethyl caprylate is kind of this apple-like characteristic. Those are ethanol and caproic acid, and ethyl and caprylic acid, respectively.
Have you ever smelled those fatty acids?
Straight? Yeah. No. I don't want to again.
It's like concentrated goat cheese.
Yeah, remember how I don't like the goatiness from Brett beers?
Yeah, that's it.
Yeah, yeah. I know. Now, increasing these is difficult, and this is where it gets very complicated for it. The best option here often is to just increase the precursor. So if we look at ethyl acetate, the acid we're working with here is acetic acid. Now, we do know that when we increase oxygen rates to yeast cells, they do produce more acetic acid. Oxygen is actually required for acetic acid, which is one of the reasons why we typically see this increase. Anecdotally, we see an increase with open ferments, or more oxygen being provided. Ethyl caproate is produced from caproic acid, which is a fatty acid, and caprylic acid is also a fatty acid, which gives us more of that apple-like characteristic. It's really hard for us to edit these other than acetic acid. Acetic acid we can push the yeast cells to produce more of, but and that one's fairly easy, but anything else is very complicated. One thing I would note on here, and this is something you're starting to see some people get interested in, is that if we vorl off our wort to get incredibly pristine, clear wort, one of the things that we're removing, or at least we believe to be removing, is fatty acids. So if you're trying to reduce these, or you're trying to modulate them, there's an argument that if you vorl off your wort minimally, you're going to get more fatty acid content, therefore you might be able to push these up higher. Now, you're also going to produce a lot of other compounds, so I wouldn't necessarily recommend that you do that. But if you really want to try and see some weird flavors come out, that might be something you could look at. Or if you want to really try and clean these things up, really boil off your wort heavy and make more of a lager-like wort. But we'll talk more about the lager applications for that in the lager chat. Which then brings us to the other class of esters we're going to talk about, which is acetate esters. In beer, the most common acid here is acetic acid, where the acetate portion comes from. And you're probably all very familiar with some of these. So isoamyl acetate is the banana flavor or apple. Very common in Hefeweizens and sometimes wits and saisons. It's that kind of iconic banana ester. We also have isobutyl ester, which is isobutyl acetate, which is banana and fruity, and 2-phenyl ethyl acetate, which is more of a rose and honey characteristic. So we can modulate these a little bit. One of the ways we can do this is with fan. So this is a bit more difficult to do, but I wanted to kind of give you guys an idea for it. Isoamyl alcohol, which as we saw beforehand was a deaminated or amino acid that's been stripped of all its nutrients, a version of leucine. Isobutyl alcohol uses isobutanol, which is the version for valine, and 2-phenyl ethyl acetate is beyond the scope of this webinar. But there are some things we can do to alter these. It's just very inconsistent and very complicated. The most common way we can increase these, and there are ways we can increase these, are increase in simple sugars. We've seen this many times over, where if you increase your simple sugars, you get more acetate esters. Increasing the original gravity of the beer is going to increase these as well. So if I make a beer at 1060 or 50 degrees Plato versus 10 degrees Plato at 1040, we typically see an increase in esters with a higher gravity. Increase in oxygenation tends to cause an increase in this, and also a decrease in soluble CO2. Also, which would be open fermentation, would also increase something like this. Strain selection is also huge. The best option for hitting the desired ester profile is first to go with strain selection, then kind of, file it down and shape it to the perfection within process modifications. One of the questions, and I want to kind of highlight here, was about dissolved CO2 inside of wort and its impact on esters. And we absolutely do see an increase in acetate esters when we decrease the amount of CO2 inside solution. And one of the ways in which you see some brewers do this is actually by keeping particles of trub inside of their fermentations. Now, trub can act like a nucleation site during the ferment. And nucleation sites, for those who don't know, are the things you have in the bottom of your beer glass where bubbles rise up from. They're areas where we decrease the surface tension for gases to leave. So trub, by doing so, will actually knock down the soluble CO2 concentration in that substance. Trub also acts as a source of fan during the ferment. And depending whether it's hot break or cold break, that can be a good or a bad thing. Which is why it's not usually recommended. And I usually recommend that you try other sources of this first, but some breweries do that with high degrees of success. Very high degrees of success. And this is something a graph that Richard pulled up just to kind of show the increased gravity portion. So as we increase the gravity, we had the two esters here, ethyl acetate and isoamyl acetate. You can see them increasing as gravity increases. That's a pretty common phenomenon that we see kind of come across the board. So in general for esters, there's still a lot we don't understand. And if it seems like I'm kind of giving you half answers, that's because that's all I can really give you. Provide more nutrients can alter them, more fan, healthy yeast cells, more oxygen, less CO2, simpler sugars. These are all going to influence how much esters you actually crank out of the yeast strain. None of the research gives us a clear answer on this, and you are going to have to do, trial and error on this. We need more research, but I do foresee us doing a webinar on esters, just focusing on that, because there's a lot of research and a lot of confusion with this and a lot of strain dependencies associated with it. That was a lot. That's just this is going to be a whole other topic. Richard, do you have anything you want to kind of highlight?
Nope. Yeah, I mean, the other thing I would say is that that there is really not a lot of research on this, especially when it comes to the yeasts that people are using now. Especially when we're looking at home brewing and craft brewing and using ale yeasts. A lot of the research was done on lager yeasts, and the ale yeasts might do different things and might do things that are that run counter to what we've observed in the lager yeasts. And so I think with time we might see maybe some of these rules bent or upended in the future as more people start to research a more diverse set of yeasts.
Yeah, I would want to just kind of stress that a little bit. So if you look at almost all research that's been done on beer until probably say the mid 2000s, it was all for lagers. That was it, because those are the people who actually had the money to perform research. We're only really starting to see research being done on ale fermentations, which are now becoming some of those common fermentations in the last decade at the best. We are kind of behind when it comes to research on this sort of stuff. So there's probably a few things that we believe about ale at least that aren't true. We'll find them out. We'll find them out soon enough. So next one, does initial fermentation temperature impact fusel alcohols? Absolutely. Anything that increases rate of yeast growth will typically do so. So higher temps, more sugar, they're all going to increase fusels. They may not increase esters because esters are dependent on fusels. You have to have other things to get the esterification to occur. How does alcohol level affect ester perception? Richard brought this up. Alcohol is a volatile carrier. It actually helps strip aroma compounds off products, and it can help enhance aroma. This is influenced by the type of alcohol. So if we have fusels or other flavorful alcohols, those can negatively impact the esters that we're tasting. Kind of the sum of the parts being greater than the individual, so to speak. This can be negative or positive. It really depends on the situation of each individual beer. Is there any scientific research comparing open versus closed fermentations and their respective effects on flavor in the finished beer? Not really. Nothing formal. There's a lot of kind of institutionalized knowledge regarding this, but there's nothing heavy-handed being used. I know heavy research being done. As I mentioned beforehand, almost all research being done is focused on macro-style brewing. And most of the research being done now is not using kind of modern techniques. Sorry, it's all using modern North American craft techniques, which open fermentation doesn't quite go there. So when you see things come out about open fermentation, it's usually institutional knowledge that breweries have found it themselves and things that worked well for them. They may not understand why it is, though. So there's a little bit of actually a quite large knowledge gap in there. Yeah. And then we'll move on to yeast repitching. So this was a fun topic, and I had some. We are going to delve a lot more into this topic when we talk about yeast management, the kind of the fifth section in this webinar, so to speak, because that's going to be a whole one. But I really like this question. So how to increase viability and vitality when repitching English ale strains and IPAs? Now, for the home brewer, this probably isn't something you're worried about. This is definitely just a pro brewer thing. I want to kind of explain why this can be a problem. So most of the yeast that are being used for hazies, for New England IPAs, they don't flock well. They do not flock well almost at all. So when it comes to trying to repitch them, this can become a problem because you often won't get enough. I shouldn't say often, but it's a possibility if your technique isn't right that you won't get enough yeast cells off of that batch for you to make the next batch of beer with. So making sure you have techniques to harvest the proper amount of cells is really important. For home brewers, you can just buy new yeast; it's no problem. But for pro brewers, where it's not a possibility, you have to repitch. This becomes a much bigger issue. Additionally, from a pro brewer standpoint, you never want to be pitching yeast that contains hops in it. Hops are toxic; they're poisonous to yeast cells. It's not good, not poisonous, but they're damaging and stressful. It's not something you want to do. Now, we are going to address this a lot more with more pictures and things of that in another webinar, but we'll give you kind of the Coles notes now. So there's two main options. You can either harvest cells before the dry hop, or you can move the beer into a secondary tank that has the dry hops already in it. So harvesting yeast cells off your main tank becomes a breeze. So the first method is to kind of ferment as normal. Many brewers will then do this kind of pseudo crash around 15 degrees Celsius or 59 Fahrenheit for about 24 hours. And this is thought to do a few things. It will slow the rate of fermentation down. It'll also increase the solubility of CO2, so you have less bubbles moving throughout the liquid, which will mean the yeast cells have a greater chance to sit and lay on the bottom of the tank. At that point, you then harvest your yeast, which should be much easier at this point. And then you dry hop the beer as you normally would. Let the tank free rise back up if there's still some fermentation left to go, and have at it. That's the basic way to do it. And this works for, it's very easy to implement. It's a very simple approach for this. I would say you run a higher risk of this going south or just going incorrectly by doing this method. Which is why the next method is kind of what I would recommend, but it has some logistical problems to it. So method two is you ferment as normal. And then, halfway through, 2/3, 3/4 of the way through fermentation, as we kind of talked last time about the free rise near the end of ferment, when you're about to do that, you transfer the beer from tank A to tank B. And there's many benefits here. The first one is that you can actually put your hops in tank B, the receiving tank. And the benefit here is that when wort hits that, or beer at this point, hits those hops, it's going to foam like mad. But the foam in this case can expand throughout the entire tank, which makes your life safer, makes everything safer, less mess, less issues, less all sorts of stuff. You now have a tank that's full of beer that has like your dry hops nicely mixed because also the wort came in from the bottom and roused everything as it did. And you have your initial tank where you just have an easy to access yeast cake. So you can then bubble some sterile gas through it, not oxygen, use some of those other gas, homogenize the whole thing, harvest it, makes your life super easy. The problem here is that this requires you to have a spare tank. It requires you to constantly have that extra tank as the receiving tank inside the facility. Now, logistically tricky, this makes your life much, much easier. So this is something that you as the brewer have to figure out in your own facility kind of on a week-by-week basis. This is kind of a quick little rundown on it. We do have a blog post about this on our website at escarpmentlabs.com. If you want to look more into this, I believe the blog post is called A Practical Guide to Yeast Management. You can go through there and kind of get a better idea as to the two different methods and see for yourself which one makes the most sense for you. Richard, anything to add?
Nothing to add there. Yeah, the one thing that I would mention, and we'll get into this a little bit in the biotransformation talk, is that there are ways to just sort of be strategic about when hops are added, that the brewer can avoid having to do, or avoid as much as possible having to do the mid ferment dry hop that creates all of these yeast management challenges.
Mm-hmm. Yeah, yeast management with New England style IPAs is it's a problem.
Oh yeah.
Yeah, it's a big problem. So go to the next one, and this is one that's going to require a little bit of explanation. So how many generations of yeast can yeast strains be reused in home brewing before a serious mutation occurs? And this is not a simple question. I would akin this question to how many times can you drive your car before it stops working? And the real answer to that is it depends on how well you take care of it. This is an example of cell health, by the way. This is, we can keep on using healthy yeast indefinitely. There are a handful of brewers that have been using the same yeast culture, so to speak, for decades. But this is where we have to determine what's the difference between cell health and mutation. So mutation always occurs. It's never not occurring. Cells are always mutating. We are always mutating as well, by the way. Now, most mutations are benign. They either have no positive or negative impact on the cell. They just kind of happen and they're just there. Other mutations are immediately lethal. So if that's the case, that mutation occurs and the cell dies. Mutation is always going to happen. We're always going to see some shift of the culture. But whether or not it's actually going to have an impact is a whole separate question. So to give you an idea, that there was a handful of these breweries in the 1980s that opened up that used a strain of yeast called ringwood. It's actually kind of a whole system. They all use the same yeast. And this yeast was meant to be repitched over and over and over and over again, to the point where many of these breweries have been using the same yeast culture since the 80s. It's rather impressive. Now, this is where it's important to understand the difference between cell health and mutation. This strain is not going to be the same from year to year. This strain is going to mutate. The strain is going to keep on going. The strain is going to keep on changing. Any cells that did mutate to the point where it was a lethal mutation, that one cell would die, but the entire culture would keep on, I don't want to say evolving, but keep on changing. So to answer this kind of shortly, the answer of how many batches can I do really depends on how much time you allow things to happen. It's not as simple as 6 batches and our cell has mutated. Now, a logical kind of follow-up question to this would be, how do we as escarpment ensure that we don't have this happen in the cells that we're plating? And how do I know that the Vermont I'm using now is the same as the Vermont I got 3 years from now, or 3 years in the past? And the reason is that the way we get around this is we freeze all our cultures down. So we have all the cells frozen in small little vials, and this allows us to whenever we need to, go back to these vials, pull samples from there, re-streak them out, and ensure that the culture we're working with is the same genetic culture we're working with 1 month ago, 2 months ago, 3 months ago, 6 months ago, consistently several years ago. And this allows us to make sure that we're always selling the same organism. We're not selling year 3 of Cali. We're selling Cali ale. It is always the same. If you want to try, if you're keeping yeast cultures in your fridges or using them in your house consistently, they're going to be changing. All cultures do this. There's no way around it. Now, to answer the question a bit more directly, cell stress can accumulate very quickly. If you stress the cell out and the cell is not able to, it's not a very high stress capacity cell, you can take a really healthy cell population and kill it pretty quickly. You can stress that thing out. The next fermentation is going to be very poor. It's going to be very, very bad. So they're not going to produce good tasting beer. But mutation is always going to happen. It's always going to happen. There's no way around that. I do want to point one thing on here. This is more so from the pro brewer side. I remember when people were just kind of switching away from Cali to other strains, because Cali used to be the dominant organism everywhere. People started getting really frustrated with Cali near the end of, I'd probably say like 2012, 2013, I'd say. Because what you started to notice was that Cali started to become more attenuative. Your Cali that was originally 86% would now go to 87 or 88, 89. I don't believe this is a case of mutation. I would be more inclined to say it's probably a case of cross-contamination, because a lot of these facilities were also using other yeast strains inside of them. So I wouldn't be surprised if that's what was actually going on. But that's strictly a hypothesis occurring. Richard, do you have anything you want to weigh in on that one?
Yeah, there is some interesting work on the Cali strain, Chico strain specifically from Maitreya Dunham in Washington. I don't think that's been published, but something cool coming out about gene mutations and rearrangements that happened surprisingly early in that strain. So there's interesting stuff going on there.
Yeah, Cali is pretty bad at being a yeast, but it's really good at being a brewer's yeast. It's kind of fascinating. It's very hyper specialized. So yeah, keep going. This brings us to the last topic, which is yeast propagation. I had fun with a few of these questions. One of them is a little frustrating though. So question 1, can you explain the 10-to-1 yeast growth ratio and why that's recommended ratio for growing up yeast? I get this question a lot from my students, and this is somewhat incorrect in regards to that you have to follow the 10-to-1 rule. The 10-to-1 rule has a lot more to do with hedging your bets and a lot more to do with what you should do. So the 10-to-1 rule kind of skirts a large amount of potential error. And I try to describe it like this. So if you have one contaminated cell per 100 yeast cells, that's just over a 1%, so just shy of a 1% error. That 1% error is not going to be outcompeted, which means that error cell is going to keep on accumulating, growing and growing and growing and growing. Now, if you have one contamination cell per 10,000 yeast cells, that yeast cell is going to outcompete it. The chance that one bacterial cell, or whatever it may be, is likely going to get starved out, or it's not going to have enough nutrition for it to keep on going. So this is the idea behind it. If I go a small amount of yeast in a big flask, I am pushing myself closer to that 1 in 100 scenario, because my yeast to potential bacteria or contamination ratio is a lot larger. But if I constantly go 10-to-1 increments, like I start with 10 mils, and I then go to 100 mils, and then I go to 1,000, so 1 liter, 1,000 mils, I am going to ensure that I'm always adding a high amount of yeast cells to it, so my chance of having contamination is going to be less. Now, we don't operate this at the lab. We have better training, better technologies, better equipment, so we don't have to do the 10-to-1. The 10-to-1 on a commercial scale doesn't make a lot of sense. If you have to use 10-to-1 on a commercial scale, you're doing it wrong, or you don't have the proper training. But from a home brewer standpoint, I would heavily recommend the 10-to-1, just because it's going to make sure that any errors you have don't accumulate. Anything to add?
Yeah, no, I would just add. Maybe there's a few strong opinions here. I would say that most of the starter calculators that are out there are wrong. They will tell you to pitch way more yeast than you need in your yeast starter. If a starter calculator is telling you that you need to pitch, 2 pitches of yeast into your homebrew sized starter to get the yeast you need, look at who wrote those calculations, and you might be able to figure out why it's telling you that. I would say you almost never need more than 1 yeast pitch, regardless of your starter size. If you're under 5-liter starter, to get the cells that you need out.
Yep.
And then beyond that, like again, there really isn't necessarily a lower bounds. It just comes down to the quality ramifications. The local macro brewer starts with I think a 1 liter pitch of slurry into their 20 hectoliter propagator. And, it works for them because they've got a lot of quality control measures in place. They know it'll take 72 hours for the culture to grow, and it works out for them. So it really just comes down to how confident you are in the quality control.
Yeah, one thing, Richard, I also mentioned, this is someone put in the chat, that is Brewkaiser okay.
They're all wrong.
Yeah, exactly. It's not. I think they may have put a little too much onus on the companies themselves. I would say that the calculations here are wrong. And one thing that the calculators also get really incorrect is that they assume that almost all yeast cells grow the same rate, into the same density, and to the same everything. They assume that a Saison yeast is going to act the same as a lager yeast, is going to act the same as a New England IPA strain. It doesn't work that way. Believe me, we grow a lot of these things. They don't grow that way. You need to kind of do a bit more to it. Now, I would, as you kind of mentioned last time, I would recommend you overgrow cultures and undergrow cultures. So if, say, you go through a calculator and it says you need 2 pitches, I would probably use 1. And if it says you need to make a 1 liter starter, I'd probably make a 2 liter starter, just because having more cells isn't ever a bad thing. This is part of the reason that why we try to give you guys more yeast cells in our pitches. And then that's just kind of a personal preference for us. We don't want you guys to have to do yeast starters, because I think we've all banged our head against the wall enough, or even with a microscope, to try and figure this stuff out and realize it just doesn't go as simply as you think. We haven't published, Brendan, our growth data. That's something that we kind of keep a little, I don't say close to the chest, but the nutrients you use and supply to the culture are incredibly important. They radically improve growth, and that's the part that if we did publish our data, it wouldn't be exactly useful to people, because we couldn't tell you our medias and things like that. That part we play very close to the chest.
Yeah, I will add that building a better calculator is something that we've always been talking about and always backburnered, and now that there's a little bit more work at home time, I think that is something we will put some energy into and making available to people. And the main goal would be let's make a calculator that focuses on the yeast strain, because they all produce different outcomes in terms of cell density. And the amount that we start with is totally irrelevant, and we have data that can back that up. And we'll definitely be tooting that horn when we have that available.
And when we first started the lab, we were pretty focused on this. Like, we were trying to, do we add 1 liter of culture or 2 liters of culture to get this propagation going? And
it doesn't matter.
It doesn't matter. Looking back on it, that was silly. We've gone through all the frustrations of trying to use these calculators to validate our own work, and it does not work. It doesn't work. But more cells is more, so I would recommend to do that. So question 3, if FVs play a role, fermentation vessel play a role in different yeast characteristics, do you prop different yeast strains in the lab in different shaped vessels? So it's a 2-part answer. Yes, we do prop them in different vessels, and we do have different size vessels for growing certain strains. Like Saisons have a wider tank, other cells have thinner tanks. This is a look at a few of them. They are very different, and we do preferentially grow certain cells in certain tanks. Just so we can, probably maximize yields and things like that, maximize health and cultures and all that good stuff. Yeah. Anything to add?
Nope. Man, I missed that place.
That place? Yeah, we haven't been. Yeah. The whole quarantine not being there every day is it's weird. It's very weird. I don't like it. So question 4, what's the recommended way to make a starter with a stir plate? I love this question. Ideally, you have an autoclave. If you don't have an autoclave, I didn't put this in here, an Instant Pot does have a pressure cooker in it, and that works really well. I know a handful of breweries that are doing that as kind of small scale stuff. At home, boil on a heat stable flask on the stove with tin foil on top, let it cool down. I'd make that media with 140 grams of dry malt extract per 1 liter is kind of where I usually start it with. Add a few hot pellets in there or two just to give it a little bit of IBU. Stir until it starts to flock or foam subsides. Remove it from the stir plate after about 2 days. Remove it once it's about 2/3 away through its growth, and then put it on the counter for about 2 days. If you don't have, or you don't want to do gravity readings with the starter, which is what I would recommend you do, and it's what we do for our propagation, 36 hours or until you notice it's quite cloudy, let it sit on the stir plate until that point, then take it off and let it sit. I would then place it in the fridge and then use when you want. And this is a perfect setup or segue into the next question, was essentially, should you cold crash and decant your starters? And I absolutely would. I would recommend you cold crash your cells with the spent media. That spent media doesn't taste great. Like, it doesn't taste horrific, but it doesn't taste good. So yeah, if you were able to remove that, it's going to make your beer taste better. It's not required, but it will give you more consistency and it will make your life a bit easier. There's also some arguments that a cold shock will help the yeast cells in regards to kind of its next ferment, but that's a whole area, and I don't believe there's a lot of hard research on it. Do you have anything to add?
Yeah, I think there are some benefits to cold crashing yeast, specifically because it does help them to build up the reserves of storage carbohydrates like glycogen and trehalose, and that can actually be helpful for the yeast when they then go back into growth and fermentation. So it can be beneficial.
Yeah. And there's it's really hard to kind of reason this data out, but we anecdotally see some benefits from cold crashing too. So I would encourage you to do so. And with that, that's back to you, Richard.
Excellent. So we're going to talk a little bit about yeast nutrition. This was an awesome question because I didn't know anything about it until that question was asked. So it forced us to kind of dive deep into understanding this, because I didn't know that zinc affected flocculation that much. So it's well known that calcium is a major driver of flocculation. Like, if you have more calcium in your water, that's usually going to help with yeast flocculation. But yeah, zinc can also impact flocculation. It does seem to be strain specific. So this is some research from 1990 showing that two ale yeasts do respond to zinc, and it does help them with flocculation. But then you look at the right on the lager yeast, and nothing's happening. So it seems like ale yeasts respond to the presence of zinc, and that helps them with flocculation, and lager yeasts don't. So it might come down to genetic differences between those yeasts. Maybe the eubianus contribution to the lager yeast has changed how it responds to zinc. Next slide.
Yep.
I'm just going to keep diving into this because this is something that I think is cool, and I'm going to try to make it cool to everyone else, I hope. Because I was starting to think about this question. I'm like, why would zinc impact flocculation? What the heck's going on here? And it doesn't really seem like it's ever been like really, really well studied, to be honest. One thing that is known is that the flocculation genes, the flow genes, those are partially regulated by this guy called MIG1. MIG1 is like a master gene regulator. MIG1's got its fingers in every pie. When we look at yeast metabolism, it's doing a bunch of things. One thing that we know about MIG1 is that it needs zinc. It uses zinc as a cofactor to help it bind to DNA sequences and repress genes. So given that it needs zinc, maybe zinc impacts the function of MIG1 in the lager yeasts and the ale yeast differently. So for example, if you look at the bottom here, we can see another study where if MIG1's been removed, the yeast actually doesn't flocculate. So clearly this has a lot of control over the flocculation of the yeast. So maybe also if MIG1's present but doesn't have enough zinc, then maybe also it still is not able to control genes and make the yeast flocculate. So maybe that's what's going on here. I think that's very interesting. It's something that a number of us at the lab have actually studied in the past. And so, yeah, thank you for humoring me in this sort of nerdy detour. But I think this is cool stuff because it sort of highlights like how complicated yeast can be and how, but also how intertwined all of these regulation things are. Like MIG1 regulates how sugars are metabolized. It controls which sugars are consumed in which order. So it's doing all of these different things that are relevant to the yeast and its fermentation. Go to the next slide. I just want to highlight that's important, getting nerdy about this, because one day there will be a paper out on MIG1 from a few different escarpment folks, including our other co-founder Angus from back in our research days. One day that'll happen. So that's exciting. If malt supplies enough nutrient, why do we consider adding yeast nutrients? So usually malt does supply enough fan, especially if your wort is over 1050 in general. Fan supply is proportional to the amount of malt that we've added. But one thing that I think is important to mention here is that malt doesn't always supply consistent amounts of the micronutrients. So things beyond the fan, like the vitamins, our B vitamins like biotin, thiamine, pantothenate, are important for yeast nutrition as well. Some yeasts need more of these vitamins or less of them. So just to provide one example, we've found that Vermont really needs biotin. We almost treat it as like a canary in the coal mine for nutrition, where every time we're getting in a new lot of malt, we test that malt on Vermont. And if it doesn't grow well, then that's a good predictor that that malt is deficient in micronutrients and that we need to make adjustments to the wort production in order to ensure that we have consistent propagations. So that's something where we've been able to find some interesting nutritional quirk, and it's been able to help us with consistency in the yeast lab. Yeast nutrient, like commercial yeast nutrients, like Yeast X and Fermate K and stuff like that, they typically also supply useful ions like magnesium. Magnesium helps with alcohol tolerance, so it's a really useful one to supplement yeast, especially for higher gravity brewing. And then, of course, zinc, just like we've talked about, it helps with flocculation, also helps with fermentation. It's a pretty important one. So malt supplies enough of the macronutrients most of the time, but for some yeasts, in some situations, it's helpful to supply extra micronutrients that will help the yeast to ferment cleaner or ferment better.
Just a few things to add on this. When you say micronutrients, that this is mainly regarding vitamins and things like that, not just minerals.
Vitamins and minerals, yeah.
And it's the other thing to keep in mind is that this variation in micronutrients isn't. It's not very well understood. Is the first part. And the second part that we do know that certain types of barley, or anecdotally, we've been told that certain types of barley in certain regions of barley and certain years of barley have higher or lower amounts of these different nutrients. We don't know exactly the reason why this is, but as a generality, English-style malts seem to have a much higher concentration of the nutrients that are required for, say, Vermont. Pilsner malts tend to have a lower concentration of these vitamins. We're not sure why, but this is part of the reason why. Part of the thing that we've noticed. We don't know whether this is a malt house thing or whether or not this is a substrate, like the barley that's used to produce the malt thing. There's still a lot of questions when it comes to that.
In the case of a high ABV brew for which you keep adding sugar during fermentation, should I also be exposing the wort to oxygen? So the answer that I usually give is, you wouldn't typically do this. Adding oxygen forces the yeast to pretty radically shift gears in terms of metabolism, because now that it can do the aerobic respiration, it can get energy that way. And so it's going to do that. And that may actually slow down its ability to then go back to the anaerobic fermentation afterward. So for high gravity brews, we typically recommend a lot of oxygen up front while the yeast is still in growth phase. So one thing a lot of people do is, oxygenate the wort, pitch the yeast, wait 12 hours, and then oxygenate again. And for most yeast, you're still, well within the growth phase of the yeast. And this is not really detrimental to the beer because the yeast is going to take up the dissolved oxygen so fast that you're not going to oxidize any of the components of the beer. So I've seen this as a very common approach for people making imperial stouts and barley wines. I don't really make those beers very much anymore, but I know, when I did, it was common practice to, use pure oxygen right at the start, usually using like Cali Ale or Anchorman Ale, and then add oxygen 12 hours in as well. That really helped to ensure a clean and relatively fast ferment without too many off flavors.
Yeah, no, that makes absolutely. Yep.
Great question here. I've heard of people putting olive oil in their fermentation. What is the purpose of this? In theory, olive oil can circumvent the sterol building that oxygen helps with. The reason for this is that olive oil is rich in the 16 carbon and 18 carbon fatty acids, which are what the cell is building for its membranes in the first place. That's most of what the yeast cell membrane is made up of. In practice, this technique doesn't seem to be very widely adopted, although there was a study on it that was conducted at New Belgium. And one thing I wanted to flag here is like the amount that was tested was tiny. Like the amount that you need to supply the yeast is tiny. It's like the head of a pin basically in a homebrew batch. So it's just something to flag there. Next slide.
One thing to add to this, and this is part of the reason why this hasn't seen a large amount of adoption, is that you're kind of doubling down on something. So one of the precursors for E2N, one of the papery off flavors, is linoleic acid, which is one of the main oils that we see inside of olive oil. So yes, if this is possible for us to not have to add oxygen to our beers by supplying them the yeast cells olive oil, but because you have more precursor that's going to be used to produce that papery compound, if you do get oxidation, that papery compound skyrockets. So you're making something easier in the brew house to make something a higher risk later on. And that, from a commercial standpoint, isn't the best approach. I know I've heard that there's a few breweries in California that are still doing this and using it kind of as their standard method. I personally wouldn't recommend it, just because there's a lot of complexity to it that you can very easily hurt yourself.
Yep. Next slide. And this is, I think, really important to flag, because I think a lot of people have taken home the wrong message from this study. I think a lot of people have heard the message that, someone tested out olive oil at New Belgium and it worked the same way as oxygen, and so you can use olive oil. But if you go and you look at the study, which is what I went and did this week, they don't say that. They say that adding olive oil to the yeast showed consistent, complete fermentations of acceptable flavor and improved flavor stability. However, the fermentations were slower and produced more esters compared to the controls. So it's not the same, and they state that pretty bluntly. So this is, I guess, a little bit of a myth that sort of floats out there, or that this study is a little bit misinterpreted.
Yeah, There's. I saw this question, and I was glad I wasn't the one answering it.
And one thing I just wanted to add here is that we've done some testing internally. We did a project last year focused on improving shelf life in lager yeasts, and one of the things that we experimented with was dosing pure linoleic acid into the lager yeast, either in propagation or fermentation, to improve the shelf life or the fermentation quality, and we never found any significant impact whether or not that was added to the yeast. So we didn't see much support for bothering to add the pure fatty acids to the yeast.
And that's about it for today. We do have a handful of questions in the chat, or in the poll. So do you want to start just going through those? We've been at it for about an hour and 18.
30 questions.
Yeah, we got a lot.
Yeah, let's start looking at these. The first question is about oxygenation and the dry yeast, and so that was sort of one of the statements that was made, is that Fermentis says that oxygenating dry yeast is not necessary. There may be some differences. I wouldn't necessarily call us experts in that arena.
No, I can't say anything about Fermentis, but I can speak to Lallemand, some of the Lallemand products, and just because that's what we use at the college frequently, because we have a ton of students who don't know what they're doing yet. So we give them liquid yeast that can be very expensive and error prone. It makes a lot of sense. So just because the pouch says you don't need to add oxygen doesn't mean that the beer is not going to benefit from oxygen. Like, the culture will still be happy. You won't necessarily get off flavors. It won't do negative things without oxygen. But that doesn't mean that the beer won't be benefited by adding oxygen.
Mm-hmm.
That's something that I have really noticed with those. Like, when students do make beers where they have aerated it, the beer is often cleaner, it's brighter, it's more, it shines. Where if you kind of don't aerate it, you tend to get more fusel, it tends to be more dull. The yeast, it just doesn't. It's not nearly as good. Like, I found oxygen to be beneficial for those beers, even though it's not necessarily needed. But that's strictly anecdotal.
Yeah, fair enough. Our top voted question now is about bioharvesting and capturing wild yeast. And just one thing I wanted to mention here is that we are going to schedule a whole webinar on yeast wild captures and really run through the methods that we use to capture wild yeast
Yep.
for ourselves and for our clients, and how we've sort of been able to do as much as we can to guarantee success with wild yeast capture, which can be a pretty inherently
Frustrating.
unpredictable process. So I just wanted to add there, like, we can't get into that right now in detail, but we're going to have a whole webinar on this. And I think it's a cool topic because, in the last year, even just with our wild capture projects, we've been finding a lot of really cool stuff.
We have.
Smelled a really awesome aromatic beer with a blend of Saccharomyces and non-Saccharomyces yeast from our homebrew guy Jonah the other day, and yeah, there's a lot of potential out there.
Yeah, we'll keep going on. Keep going down. Sorry.
Absolutely.
Do you think yeast nutrient is useful for 100% non-adjunct beers? It's always helpful. Always helpful.
Always helpful. I mean, I think there is diminishing returns, and like certain nutrients can be a problem if there's too much left over, because it opens up the beer to risk for microbial spoilage.
Yep.
So there are some potential issues, but more is better than not enough.
Adding like the teaspoon or half a teaspoon it recommends for a 20-liter batch, like it costs you pennies, and that's not going to overdo it, which is kind of the next question we have here. Is there too much you can possibly add?
Mm-hmm. Yeah.
If you stay within the manufacturer's recommendations, I don't really think there is.
Yeah.
Should wort oxygenation be adjusted to its strength? What would be the best way to check that you are actually oxygenating the amount we aim for? Ooh.
I always suggest it's the same as, if you're able to measure the oxygen, the same as the yeast pitching rate, right? If you're pitching your lager yeast, 1 million cells per mil per degree Plato, then add 1 ppm oxygen per degree Plato. If you can't measure your oxygen, which most people can't, I would say more is better than less.
Yeah, I would. This will be something we talk about later, but there's a few different issues I've seen breweries have with this. One of the biggest ones is oxygen tanks have a pressure gauge, and then you can often then downstream add a flow meter. So if I have, say, 10 psi coming in from my pump and 10 psi coming from my aeration tank, I'm actually not moving oxygen because those pressures are meeting and they're not flowing. You need a flow meter on the outside of your aeration line. Otherwise, you don't really know what you're working with. The pressure isn't useful. You need flow. And as I mentioned last time, making sure your aeration stone is properly cleaned, and I mean heavily properly cleaned, that'll ensure that you're actually getting proper aeration inside of your vessel. Because those are the two places where I see most breweries have issues when it comes to aeration.
Yeah, that could be a whole webinar, and we've been pitching that idea actually, because there's even just the mechanics of oxygenation and making sure that you're getting what you think you are into the wort is super important, and there is some nuance there.
Yep. Related to temperature shock of yeast cells, for bottom fermented beers, wort is sometimes casted at higher temperatures than the fermentation temperature, which is set lower the day after. This seems contradictory. Is it good practice though? So you're casting out wort higher than the temperature you want to ferment at.
Depends on your tank.
What do you mean?
So the risk you run there, if you're casting out at a higher temperature than the set point of the fermenter, is that you risk overcooling. One thing we've seen is that, especially with newer breweries and tanks from China, sometimes you don't always have independent control of your yeast cone, of your tank cone and your upper jacket. And what that often results in is that if something goes in at 14 C and it's set to 10, cooling kicks on because there's more surface area to volume in the cone. The cone is going to overcool, and we're looking at a bottom fermenting beer where that's where the yeast is sitting. What you can do is you can really stall out the yeast inadvertently, and most of the times if we're troubleshooting a slow lager, it's traced back to that kind of situation. So I would be careful. Yeah,
I would never recommend you do this, just because for Richard's point and for a few others, I can only see this making your life more frustrating.
Mm-hmm.
Now, if you're in the position where you physically cannot cool out your wort cold enough for lagers, and I've seen some breweries do that, where like they'll cast out wort into a tank at 16, and like I'll crash the tank essentially to like 12 or 13 over a day period, and then add yeast. That's a possibility, but I wouldn't want the yeast to be experiencing that cold crash. I'd want them to be added at the later point. And the same as aeration. I want like aerate the tank and then add your yeast cells in and go at it that way.
Mm-hmm.
How much total yeast bacteria strains do we have banked at escarpment? Oh, you answered over 1,500.
Yeah, right. we just cracked 1,500 now, so quite a lot. I'm a hoarder, but only for tiny things. So I'm glad that's the case. It's not newspapers or books or tigers. So.
As a home brewer, I have limited fermentation temperature control. I've got a lager going with S23 in my cellar at 13C to 14C, depending on time of day. I do have lagering facilities once I've done a diacetyl rest and fermentation is complete. Is this fermentation temp that big of a deal?
I think that sounds okay. Yeah, making do with
good.
what you have, right? Yeah.
Absolutely.
Yeah.
Yeah. Has there been any research into water profile affecting yeast behavior? Is that fairly neutral?
I think we've covered some of that, right? Like with the impact of calcium on flocculation, magnesium on alcohol tolerance, zinc on the flocculation as well. So, anything to add there, Nate? Buffering maybe.
Yeah, like carbonates, you absolutely would. Just because the carbonates are gonna influence the overall pH of the beer and influence how easy it is for the cell to maintain its environment. Yeah, not massively. The beer's going to change a lot based on mineral profile, but the yeast cells aren't going to be as impacted as one might think. Yeast cells are pretty hardy. They're able to handle a whole bunch of different environments. I think sugar concentration is going to be a larger impact for it and type of sugar, but that's a fruit thing. Yeah. At what amount do you reach diminishing returns when applying dry hops? I think that one's best answered in the biotransformation talk.
Yep. Yeah. I think one thing to add, I think there's been some research from Oregon State University on this specific topic. I don't recall the specific number, but yeah, we can get into that at a later date for sure. Because some brewers do add too much.
Yeah, some brewers absolutely do add too much. The other things I would look at is one of the best ways to get more out of your hops is to control oxygenation like a madman. Just make sure nothing is hitting those. Like one of the reasons why you see brewers adding hops for biotransformation during fermentation is partially because of the fact that it helps scrub off all the oxygen that's present. So you don't run the risk of oxidizing your beer, and hops are incredibly sensitive to that. One of the things that Shellhammer from the Oregon State that Richard mentioned found was that if you say I had 100 grams of hops I'm looking to add, if I did them in 250-gram pulses versus all at once, the 250 grams showed a small increase in overall flavor and preference. So there are some things like that in regards to how you do it. For those who are commercial brewers, there's something else you need to be aware of when it comes to over hopping, and that is that every single hop you add takes away a large amount of volume because that hop has to swell as soon as it hits liquid. So if you're trying to make a beer that's a bit more economical, so to speak, you want to try and get the most out of your hops because that means you can add the least amount of hops to it and have the least amount of losses. Like I know some breweries where buying a centrifuge is an order something that's hundreds of thousands of dollars to do. And they've made the ROI, the return on investment on those things, like 6 months, because where before they got, say, 12 kegs out of a batch, now they get 16, and that's all profit at that point. So that there's some economics when it comes to professional brewing that you really have to look at when you start looking at dry hopping beers. We'll do a few more. I think we got time for a couple more.
A few more. Yeah.
These are all going a bit quicker. They're not quite as. I think we covered a lot of the heavy questions. Do you recommend staggered nutrient addition for big beers? What specific gravity values define a big beer in this case?
One thing to mention for big beers that this terminology makes me think that it's, maybe something more in line with thinking more in line with this being like a mead. The key difference between a big beer and something like a mead or a wine is that as you increase the gravity in a beer, which is ultimately being extracted from grain, you're still increasing the nutrient content, right? As opposed to a mead where it's relatively nutrient devoid sugar that's being diluted down. With beer, the higher gravity you go, the more nutrients you're actually extracting from the grain. So in a lot of ways, I know it's kind of counterintuitive, but in a lot of ways, a lot of the big beers don't actually need a lot of nutrients. And just even speaking from experience, doing a lot of the troubleshooting with escarpment labs, a lot of times when we're troubleshooting, what we think are nutrient derived off flavors, it's often low gravity beers, like things under 1040, where we run into challenges in terms of like flavors and stalled ferments. With the big beers, and as if it's an all malt big beer, you're going to be supplying tons of nutrient for that yeast. So it's always good to have a nutrient product added because you're supplying those key micronutrients. But I'm skeptical as to whether like staggering those nutrients would make any difference.
Yeah, the biggest thing I notice for big beers is you want to keep your fusels under control. If you can do that with temperature control and proper aeration, then you're off to the races. You're doing great.
Mm-hmm.
Yeah. What does Nate drink out of the brown bottle? It's currently just water. I'm not currently drinking alcohol. That's a whole other thing. Have you seen any significant biotransformative ability from any other Kveik strains you offer? If so, which ones?
Yep. So we're definitely going to dive into that in the biotransformation talk on April 9th. We did have a blog post on the impact of pitching rate on Kveik, and we did sort of hint at some of that in there, showing that the Hornindal and the Arset were a little bit more active in some of the terpene biotransformations than the others. And then when we talk about yeast strain diversity on Monday, we're also going to show where Kveik sits on that spectrum when it comes to the terpene biotransformation. So we have some of that information, and we'll be sharing it very, very soon. And I will say that some of them, are pretty good at that. They compare to a lot of the yeasts that are being used for hazy IPAs, and then some of them are not as active. So there's quite a spectrum when it comes to Kveik, and that's kind of the story with Kveik in general, is that they're super diverse.
Think we got time for a few more?
Go for it.
All right. We're getting into kind of some more slightly esoteric questions.
Mm-hmm.
What's the simplest way to estimate the number of cells you have in your yeast slurries? There isn't. And this is something that's important to note. So saisons are a lot smaller cells than, say, Cali or Vermont. And different yeast cells will compact more intensely than others. This is going to be somewhat crass, not crass, but kind of rough. But an old kind of adage was that you needed about 1 liter of kind of like ice cream consistency yeast per hectoliter or per barrel of wort. That's kind of what you're looking for. So that equates to, what is it, about 200 mils of moderately thick slurry for a 5-gallon batch. That's probably more than you need, but that's a really rough way to do it. Richard, anything to add?
Yeah, it's so strain dependent. Like what I can say is like a relatively thin slurry of English yeast will look a heck of a lot more dense than a relatively dense slurry of saison yeast, where a saison yeast can be basically free flowing, and a English ale yeast can be basically pancake batter. And you look at them under the microscope, you've actually got the same number of cells there. So yeah, it's very strain dependent.
Yeah. I'm going to summarize this next one. We essentially is negative crashing tanks at -1°C, negative on cell health. Based on the ethanol we find in solution, it should be fine because we're not going to see any freezing occur. The density increases, so we actually see from going for like 4°C is actually the most dense water you can get. Anything above or below that, the water becomes more dense. So theoretically, sedimentation is going to occur slower, but you're working with the yeast cells. I wouldn't have any issue with -1, but I don't personally use that myself. I don't use it at the lab either.
Well, I mean, I would say that for some of our tanks, we'll go down to -1 is part of the fluctuation for sure. But like some of them we do target 0. So.
Yep. There's a question on here. Actually, we're going to skip that one and answer that one at a later point. There's one here I want us to kind of go over, and I think we'll do one more after this. In order to determine how far the yeast will attenuate, a forced fermentation test is usually done. What is the recommended time to analyze the test to be trusted? I have experience with beers attenuating further than their fermentation test, which makes it a bit pointless. Any contamination is pretty much discarded. So essentially, forced fermentation test question. The first bit I'd want to point out in here, and we'll probably do. We are looking at doing kind of a cheap like lab 101, like how to set up a lab and tests you can do. The forced fermentation test does often over attenuate, and that's for a handful of reasons that we will go past. Then the forced fermentation test really is. If you have acetobacter, you'll smell it right away. If you have lactobacillus, you probably won't get it very quick. The forced fermentation test really is there to show you whether or not an under attenuation in the brew house is due to your yeast cells or due to your brew house. Like if you under converted your grains, the forced fermentation test will not attenuate as you expect. And that means you have a brew house problem, not a fermentation problem. So that's kind of the greatest utility out of it.
Yeah, I would say beers in the tanks fermenting lower than the forced ferment is surprising. It may depend on the yeast, especially when you get into the diastatic yeasts. We've even seen in the propagations, some of the diastatic yeasts don't always ferment very far, even though they're being aerated. But then you put that yeast into a wort, and it'll ferment the wort down to nothing. So if you're using a diastatic yeast, that might be a situation where the forced ferment really doesn't match with what you see in the fermenter.
Absolutely. And this last question I kind of picked up, and I think it's kind of fun. Do we like our yeast starters shaken or stirred?
I don't think it matters.
No. And does the speed of the stirred starter impact the cell's shear stress, or does it occupy any stir speed? So essentially, does stir speed impact?
I think you'd have to be stirring pretty fast to cause shear stress to the yeast. Like centrifuges can cause shear stress to the yeast, but that's typically rotation, quite a bit faster than we might see in a stir bar on a stir plate.
Yep. So stir bars are also designed to be low shear. That's why they're smooth, typically on the outside of them. They're not meant to be a blender blade by any means or anything like that.
Yeah. Don't grow your yeast in a blender.
No, don't do that. Bad idea. So yeah, I think we'll end it there. There's a lot of good questions. I think we'll kind of try and follow up with some of these more. We might do another Q&A at some point with after we go through a few more of the webinars, try and answer a few more questions. Anything else we want to end off on?
No, I just wanted to mention that we have the next round of our webinars up on the Crowdcast page. We'll also be posting about those on social media, but coming up on Monday, we have our beer yeast flavor diversity talk. Again, if you want to know all the nitty-gritty details about all the different beer strains that are out there, that's the one for you. We're also going to be talking about biotransformation April 9th, and then the road to Lacto 2.0 on April 14th. So these ones are up. Hoping to see a few more signups. We've had almost 700 people at one point or another sign up for this series, which is that's awesome to see, and hoping to see some of you carry over to the next round of webinars as well. And I can tell you that now that we've got through the basics, we can get into some real cool stuff. So please stay tuned and come and check it out.
And for anyone who wasn't aware, these sessions are available on the Crowdcast platform. You can also find them on our YouTube page, just YouTube at Escarpment Labs, and you can find all these things are being posted there as well.
Awesome.
All right.
Thanks,
Take care,
everyone.
Stay safe. So we see you next time. Cheers.